Hello, everyone, and welcome to today's Friends Life Care Wellness webinar, Microbiology 101, Meet the Germs, presented by Linda Hnatow. My name is Gail Tamarchi. I'm the Friends Life Care Director of Wellness Initiatives, and I'll be your moderator for today's webinar. Before we get started, I'd just like to give you a few housekeeping tips just to make sure that you know how to fully participate. The handout for today's webinar is in the attendee interface, that you can just click that and download that. It was also in the email that you received an hour before today's webinar. Okay, and here's a screenshot that shows an example of the attendee interface. You should see something that looks like this on your own screen in the upper right-hand corner. And you're listening in using your computer audio by default. If it's not loud enough, check your speakers just to make sure that they're turned up all the way. And also, if you'd rather call in over the phone, just click on the phone call button and the dial-in information will be displayed. You see a question box there. If you have questions at any time during the webinar, feel free to type them in and just hit enter. And Linda will answer all your questions at the end of the webinar today. She's going to have a very complete presentation, so she might answer your question while you're, before you, right after you type it in. So we'll wait and see. So she will do that. And also during the webinar, she's going to have you complete a few polls. All you have to do is select the answer and hit submit, and we'll be able to see how you're feeling about things. So now I'd like to introduce our presenter, Linda Hnatow. You may already know her. She's a Friends Life Care Coordinator. She's done programs for us before, and she might be your care coordinator. But before joining us, Linda's work as a nurse included educating nursing home staff on infection control measures. And then prior to nursing, Linda was a researcher in molecular microbiology at the University of Delaware and at the E.I. DuPont Company, where she worked with various bacteria for over 17 years. So definitely an expert on the germs. Okay, so she taught microbiology at various levels and presented at both the local and national level. So as you can imagine, we are very, very happy to have her with us at Friends Life Care. Linda, thank you so much for sharing your expertise with us today.
You're welcome. Thank you, Gail, for that lovely introduction. So we are going to today talk about microorganisms, Microbiology 101. And, you know, the neat thing about microorganisms is that there are millions and millions of them, and they're really diverse, and they're actually, like, we don't even think about them. We don't even consider them on our day-to-day, life, but they're vital to what's going on in us, around us. They're important to the function of our planet. They are involved in ecological processes, like they take CO2 out of the atmosphere, they put oxygen back in the atmosphere, they recycle waste materials, they provide nutrients for plants and for us. And there are a lot of them, like I said, and there are actually microbial species that have not even been discovered yet. It's a pretty amazing world. So first thing is a quiz. So and I have to tell you guys that I know I have a savvy audience, and so I made these a little harder than usual. So this first one, again, go to your poll on your screen, and you will see this question: which of the following are not microorganisms? So your choices are bacteria, protozoa, viruses, archaea, and fleas. So let's see. I'm gonna kind of keep an eye on the poll, and then we'll close the poll once it looks like a lot of you have okay. So it looks like most of you have voted. So I'm gonna kind of close this and share it with everybody. Hopefully you can see the results. There we go. And the answer is indeed fleas. Now, some of you guys chose viruses, and I'm not going to say that you're wrong. What I'm going to say is that it's probably not the best answer. So I used to have to design questions, and so that was my trick, right? So you're correct in that all of these are microorganisms, and really what we talk about with microorganisms are things that cannot be seen with the naked eye. You need a microscope of some sort to see them. Fleas, on the other hand, are macroorganisms. We can see them with the naked eye. Now, microorganisms, bacteria, protozoa, viruses, archaea, They're microscopic. Some of them are single cells, some of them are multi-cells, some of them are cell clusters. For example, protozoa, these are typically single cell, and they're classified as eukaryotes. And eukaryotes, we'll find out in a couple slides, are more complex than, say, bacteria. They can be free living or they can be parasites, and they feed on organic matter, right? Debris and other kind of organic tissues. Archaea, very interesting. They're the oldest microorganisms in the world, you know, over 3.5 billion years old evolutionarily. They're similar to bacteria, but there are some major differences. Archaea are often called extremophiles because they live in really unusual, harsh environments. So they can live in deep sea vents that reach over boiling. They can live in the Antarctic where it's, you know, way below freezing. Acid, alkalines, heavy salts, deep in the ocean with the pressure, or even, you know, where there's UV radiation or gamma radiation. So, you know, if there's life out there in the universe, these guys may be the ones that are out there. So let's talk a little bit more about microbiology and microbes. So we already talked about, you know, these are very small organisms, and I'm going to actually quote Antony van Leeuwenhoek. Now, he lived back in 1632 to 1723. He was not the inventor of the microscope, but he was one of the first people to observe bacteria in using a very crude microscope. And his quote, I love his quote, he says, "I then most always saw with great wonder that in the said matter there were many very little living animalcules, very prettily a-moving. The biggest sort had a very strong and swift motion and shot through the water like a pike does through water. The second sort ofttimes spun around like a top, and these were far more in number." So he really started to explore that hidden world, that fantastic, in a lot of ways, world. You know, when we look at microbes in general, they're usually or microorganisms, they're divided into 7 types, and that's what we see here. So you can see that things like parasites, which are a type of worm, are multicellular, while our bacteria are single cellular. Each of these different types has its own characteristics. So the cellular composition is different, their morphology is different, the way they move around, the way they reproduce, and all of that helps to put them into these different categories. Now, some of these guys are beneficial and some of them are not. But I'll tell you that for the most part, most of them don't affect us in any way, shape, or form. So this is actually, I talked about how you have to use a microscope to see these guys, and this is actually an electron microscope. And an electron microscope actually uses electrons to scan, and then it translates into a photo. Magnification of an electron microscope is up to 10 million times. And a light microscope, like a regular microscope that you might have used in high school or college or something like that, only magnifies about 2,000 at its upper limit. So what this electron microscope was able to see are these soil microbes from beneath a ponderosa pine. So you can see it is indeed, and these are colorized, right? They're not normally that color, but they do it for emphasis. So I wanted to show you, because it's really hard to understand the scope of these guys, how small they really are. And so I found this is pretty cool, from Cells Alive. And you know, we're looking at a ragweed pollen, this green kind of bumpily thing. I think I made up that word, but anyway. And I want to start out, actually, I want to back up a little bit, because that pollen size is about 1,000th the size of the head of a pin. The head of a pin is about 2 millimeters. Now, a millimeter is 1/1000th of a meter. A meter is about 3 yards. So we're talking about, you know what a head of a pin is, right? This is 1,000th the size of it, and it would be about 20 to 35 microns or micrometers. And microns are 1/1,000,000th of a meter. And so we're going to zoom in a little bit more. The next biggest thing we see here is this purple thing, and that's a lymphocyte. So that's part of our immune system. It's a cell of our immune system. But let's look at the red blood cell. So here's our red blood cell. And a red blood cell is about 70 microns. So 70 millionth of a meter, right? And then kind of around that same size, you see this thing, and that is a yeast cell. Oh, I skipped ahead. All right, well, so the yeast cell pointed out over here is what you would use as a baker. It's a single cell of yeast, and it's 5 to 10 microns. So 5 to 10 millionth of a meter of those 3 yards. And then this thing that I jumped ahead to, the E. coli, and you can again see the scale here. Now we're going down a magnitude to 2 microns. And if you look even below that, you see this purple thing link here. That purple thing is an Ebola virus. So again, you can see the perspective. We've gone down a magnitude. Ebola virus is about 140 nanometers long, and nanometers are 1/1000th of a meter. Now, if we're thinking SARS-CoV-2, which causes COVID, that is pretty similar in size to the Ebola. It's a different shape. It's a sphere. Unfortunately, my animation got ahead of me. So this is kind of what the size of that sphere would be versus the Ebola virus. So we'll just skip right ahead here to rhinovirus. So rhinovirus is the smallest thing we have on here. On this picture, you can't even see the rhinovirus, but it's like right in there. So it's another magnitude smaller. Rhinovirus is about 30 nanometers. So here's our Ebola virus, this purple thing over here, and you can see how tiny the rhinovirus is. Rhinovirus causes colds, one of the things that can cause a common cold. So we see that, there's really, a vast difference in how tiny these things are. And this is just one way for us to visualize the smallness of these microbes. Now, it's estimated that there's over 1 trillion different microbe species in the world. And this is including everything, bacteria, yeast, the whole, everything. And just to kind of put that in perspective, if you take a teaspoon of soil, there's over 1 billion individual microbes in a teaspoon of soil. But today, we're going to focus mostly on bacteria, viruses, and fungi. So bacteria really are single cells, and they reproduce by what's called fission. So they split apart and they become 2 equal, bacteria cells, basically. They're pretty simple. Fungi are a little bit more complex. They're eukaryotes, and so eukaryotes we're going to talk about in the next slide. So I'm not going to jump ahead, but they're pretty much, we're going to talk about yeasts and molds. Yeasts are single cells, like our baker's yeast. It's a single cell. Mold is filamentous and can produce spores. So if you look at this, this is actually an agar plate, a piece of an agar plate that we're looking at. And these are molds. So you can kind of see that they're filamentous, they're different colors. And then these guys here are actually bacteria colonies. So they're not individual bacteria, but it's millions and millions of bacteria that are all living together in a kind of commune setting. Viruses are submicroscopic. They're really, really tiny. They don't really have a cell. In fact, the question of whether they're living or not has often been brought up. Viruses are also unique in that their genetic material can be either RNA or DNA. Now, DNA is deoxyribonucleic acid, and that's what we have. We have DNA as genetic materials. DNA fingerprinting you might have heard of, or maybe you even had a DNA test. Viruses can have DNA, but they can also have the So for us, DNA starts out and it gets transcribed into RNA, and RNA is then read by the cellular machinery in our cells to produce proteins, which become the building blocks. Viruses can also just be RNA. For example, coronavirus is an RNA virus, right? Viruses need a host. So when we talk about, I talked about eukaryote and prokaryote, I just kind of want to give you this schematic. So bacteria are prokaryotes. It's a simpler cell. One of the hallmarks is that the nucleic acids, the genetic material are not really in any defined space. On the other hand, eukaryotes are actually much more complex. So you see there's a lot of these little organelles in here that have specific functions to help the cell live and grow and divide and all that stuff. And the genetic material is actually inside a kind of sac called the nucleus. So remember, fungus or fungi are eukaryotes, and our bacteria are prokaryotes. So here's a bacteria. And this is just another way of looking at another cartoon. Viruses, on the other hand, are even simpler. They have the genetic material inside what's called a nucleocapsid, so a very simple kind of container. And then that is really surrounded by an envelope. So they're kind of in this sac, and then you may also see these kind of things sticking out. I liken that to Velcro because it helps the virus to stick to things and also to sometimes get incorporated into cells. We'll talk a little more detail on that. We're going to start with fungus because I like to consider myself a fun guy. I have to put that one in. Come on. I could also say that this is actually a pita that I found in my kitchen, I'm sorry to say. On the bottom of it, unfortunately, I didn't get to the pita in time, and I did a science experiment. There were now fungus among us, right? Sorry, I had to put that one in too. But the neat thing about it, and whether you go gross, this is really gross, or you go, this is really cool, because you see the different morphologies, the different colors, the different textures. Fungi, the kingdom fungi, so the big classification fungi has a lot of variety in it. And the interesting thing is that scientists probably have identified about 100,000 different species of fungi, but it's believed that there may be up to 1.5 million species on Earth. We just haven't discovered them yet. So fungi really encompasses everything from mushrooms, yeast, black mold, and even the mold that produces antibiotic penicillin, right? So they're all part of that big classification of fungi. They are eukaryotes. You know, originally it was thought that they were plants, fungi were plants, but they don't do photosynthesis. And actually the fungus cell is much more similar to an animal cell. Fungi in general are opportunistic, so they can get their nutrients from a wide variety of sources, and they live in a whole bunch of different environmental conditions. They're also known as heterotrophs, and heterotrophs are organisms that get their nutrition from complex organic substances. Fungi often will be decomposers, so they're important to the environment. You know, they produce these external enzymes that break down the organic matter into smaller molecules that the fungus can now use as food. They have a rigid cell wall made out of chitin, and they also are very complex. They can live in a symbiotic relationship with other organisms. And symbiosis just means that they're mutually beneficial, right? So they're going to interact together. Sometimes you have fungus that live in the soil that help plants to fix nitrogen. Lichen is a great example. So here's a picture of a lichen. A lichen is an association between fungus and a photosynthetic partner, usually an algae of some sort. And they're kind of cool looking too. Fungus can produce spores, and this is where maybe humans have a negative interaction, because those spores can actually cause irritation to our lungs or to our skin, especially for people who have weakened immune systems. In general, fungi are classified into 3 different major groups. So there's the macroscopic filamentous fungi. So we're talking about mushrooms here. You know, and then the ones that we're going to focus on are the multicellular filamentous molds. And their really main thing is that they break down organic matter in the soil and in other things too, not just the soil. Single cell microscopic yeasts are the 3rd type. And so these we talk about, you know, for me, as soon as I think yeast, I think beer and bread, right? But any kind of fermentation, really, yeast can be involved in. The other interesting thing is, remember I said that because they're eukaryotes, they're more similar to animal cells than they are plant cells. And because of that, yeast such as Saccharomyces plays an important role in research, actually. So especially cancer research, so they can use these cells to look at what's going on inside the cell. There are other types of yeast that are opportunistic pathogens, and opportunistic pathogens are ones that normally are there. They don't hurt anything, but if the balance gets upset, the environment changes a little bit, now all of a sudden they can cause infection. And a good example of that is Candida. So Candida can cause yeast infection in the mouth. It can cause urogenital infections. And that's because the balance has gotten upset. So again, molds and yeast. Yeast are single cells. They can live in the presence of oxygen or no oxygen, but they have a narrow range of pH. So they're, you know, they have to have a pH balance in order to live. They're used by us in baking, food beverages, alcohol production, and they can stick together. So even though they're single cell, they may kind of form this, what's called a pseudohypha, and I'll show you a picture of that on the next slide. They have been implicated in weakening the immune system and contributing to things like asthma and Crohn's disease. And so here's a good use of yeast. This is strawberry wine fermentation. Looks pretty good, actually. And then molds are multicellular. They need oxygen to live, but they can live in a wide pH range. And because of that, they're actually implicated, or they're actually not implicated, they are known to break down food stuff because they can live in this wide pH range. They do have filaments known as hypha, and they can divide either asexually or sexually, where there's an exchange of genetic material. Really, that's what it means in this sense. We do use them in cheese making. They can cause respiratory infections and allergies. And so this is actually a cartoon of that sexual versus asexual reproduction. So sexually, 2 different ones come, they exchange genetic material, and now you have a different kind of a mole, they both go asexual, of different kind of a fungi. They're both sexual, asexual. With the asexual, it's basically they form these kind of little tiny little things, spores in a way, and those will then develop into full-blown fungi. So this is a picture of the Pseudohypha, and remember that the Pseudohypha is the yeast. So they're not really, normally yeast is a single cell, it's kind of oval shaped. That's what you see here in this micrograph. This is actually a mold. And so this is true hypha. And so this is budding, and it's gonna, those little spores are in there, they're gonna release and form into a full-blown mold. So with humans, you know, because we're always interested how it interacts with us, normally fungus of any kind are really held in check by our immune system and our normal flora, because we have bacteria, yeast, all sorts of things on us and in us. But say, for example, you take antibiotics, all the bacteria are destroyed, the balance gets upset, and now the fungus can grow unchecked. And so that's what you see over here. You can also see it in things on your nails, you can see it on your skin, you can see it in your mouth and throat. That's what we're seeing over here. So this is the yeast Candida, and it can form, it's normally in your mouth, but say you took antibiotics, now all your bacteria in your mouth are gone and the Candida takes over and you end up with what's called thrush. Sorry if I'm grossing anybody out, but it's, you know, it's, yeah, so there it is. Yeast and fungus in general can get into the bloodstream or the lungs, and that's when it becomes really dangerous for us. So here's some more pictures of different kind of fungal infections. This is athlete's foot. It's called tinea. This is nail fungus. So you know, this is the tinea that will cause either one of these. And then this is actually ringworm, and ringworm is usually caused by a mold that becomes parasitic on the outer layer of our skin. And it can be caused by a whole bunch of different organisms, including trichophyton, microsporum, epidermophyton. And this epidermophyton also, this is a hair, a human hair, and you can see how it's kind of attaching onto the hair. Pretty cool stuff. Now we're going to talk about bacteria. Now bacteria, so this is actually an E. coli bacteria, and I want to tell you that my background, if you look at it closely, these ovals are E. coli, and then all these like stringy things coming off of them are their flagella. That's how they move around, and that's what this is. This is actually a 28-meter long inflatable E. coli sculpture that was created by an artist named Luke Jerram, and this was in the UK. The University of Oxford Natural History Museum had this exhibit last year where it was called Bacteria World, and I'm going to read you this quote from them because I thought this was pretty neat too. Bacteria survive, they thrive, fight and die by the trillions every moment. They swim using nanoscopic motors and battle with spears. They sense, communicate, remember. And as scientists discover more about these tiny organisms, it's becoming clear that the bacteria wield huge influences over us, shaping Earth's past, our present, and the future for us all. We have only recently realized how much our lives are inextricably linked with the lives of bacteria. We really are living in a bacterial world. So pretty cool stuff. Oh, so here's our next poll. So in this poll, I'm going to ask you which of the following diseases are not caused by bacteria. So Gail is going to open the poll, and you guys can pick which one this is. And wow. Okay. People are jumping right on it. I'm telling you, I knew you guys were very savvy about this stuff. Okay. Not caused by bacteria. Okay. So we kind of, I'm gonna, I don't know if you guys can see the poll results. Yes, Gail has the poll results. And actually the answer is flu. Influenza is caused by a virus. So pneumonia and UTI can be bacterial. They can also be caused by other things too. But they do or definitely can be caused by bacteria. So when we talk about bacteria, they're really evolutionarily pretty old, 3.5 billion years old. They come in all different kinds of shapes and sizes. They're very diverse. They can live in all kinds of environments, the soil, the ocean, the Arctic, in the human gut, on the human skin. Remember, they're prokaryotes. They're pretty simple creatures. Inside their cell is this cytoplasm, and that's really this kind of soup of proteins and other nutrients that they need in order to live and survive. That's the cytoplasm. And then their genetic material is here. It's in a very loose association called a nucleoid. And then they also have what are called plasmids. And this is separate pieces of DNA that are outside of the regular genome that they have. And those plasmids have extra genes on them. And I will tell you that antibiotic resistance is often on these plasmids. And that's important because when different bacteria meet up, they can exchange these plasmids pretty readily. And so that antibiotic resistance can move from not only from bacteria to bacteria, but from species to species. So they can cause some problems, crop damage, food spoilage, infection, all that stuff, but they can also be very useful. Not just fermentation, but in their destructiveness, right, they are actually breaking down substances. They are recycling. They actually drive circulation of carbon, nitrogen, sulfur, and phosphorus around the planet. And so they actually turn those elements into things that plants and animals can use. So I mentioned fermentation, things like yogurt, sauerkraut, those kinds of things can be fermented. Overall, there aren't a lot, even though we focus on the disease-causing ones. There's less than 100 species of bacteria that can actually cause infection. So they are classified. I'm not going to go through all this stuff, but they're really classified on a whole bunch of different things. Their morphology, how they react to different stains, their you can look at them under the microscope, how they grow under what conditions, what environment, their biochemical tests, what kind of substrates can they live off of, their shapes, their nutrition. So phototrophs are ones that can use photosynthesis. Chemotrophs use actual chemicals. For example, they might use nitrogen as their energy source. Autotrophs are using carbon dioxide as their energy source, and heterotrophs, like we mentioned before with the fungi, are using organic substances, usually something a little more complex. Some of them can form spores, and so they can be classified that way too. And this just kind of gives you an idea of the diversity that they have. So when it comes to their interaction with humans, there's about 10 times more bacteria cells as human cells in the human body. That's amazing. 10 times more of them than us, than our cells. A lot of those cells are found in our digestive systems, and the bacteria can be on any part of our body, including our skin, in our bladder, in our lungs, intestine, brain, everywhere. And they can be infectious in all those locations also. Mostly the beneficial guys are found, like I said, in the gut, so the gut microbiome, but there's also a skin microbiome, which is kind of interesting because we think of the skin as a physical barrier that protects our bodies from assault from organisms and toxic substances, but the skin also has all these different bacteria, and not just bacteria, they also have fungi, viruses, and mites even that live on them, and they kind of have set up their territories, and so they prevent colonization by other harmful germs. They also can change the environment so it's inhospitable to other germs. They can boost our immune system, these skin microbiome, and so can the gut, by the way. And one of the ways they boost it is basically they prime our immune system for any possible invaders. So when they become pathogenic, or when there are bad bacteria, they could produce toxins, and that might be causing the problem. They could actually invade tissues. They can trigger inflammation, and that inflammation can negatively affect our skin, our heart, our nervous system, our kidneys, our GI tract. And actually, Helicobacter pylori has been linked to cancer. So these are actual micrographs, again, of real bacteria. This one down here is gonorrhea, so we could consider that one of the bad guys. This middle one is E. coli, and you can see his flagella are really long. E. coli can be either good or bad, if we want to put that label on them. And then this is Borrelia, and Borrelia is what causes Lyme disease. It's it shows you the different shapes also of these bacteria. So the densest bacterial population on the planet is found in the mammalian gut, or in the mammal gut, according to Christoph Tang, who's a cellular pathologist. And so there's a whole bunch of them. And I wanted to show you this. This is such a beautiful pattern. This is an agar plate. Agar is kind of like a heavy-duty jello that bacteria can grow on. And this was an entry for the American Society of Microbiology agar art competition. I didn't even know there was such a thing. I wish I had. I would have entered in it because it's really cool. But this was actually, this is actually from feces. So these are the different bacteria from feces. And you can see it's actually growing on a color changing agar. So that's what gives it these different colors. And it kind of tells you the different species that are in there too by the different colors. But it's beautiful. So it kind of gives us a different perspective on those gut bacteria, I think. So this just kind of gives us an overview of what I'm calling the bad guys. So these are the bacteria that can be harmful to us. And remember that it's less than 1% of all bacteria that are out there that can actually invade us and make us ill. But there are a lot of them. And again, I'm not going to go through all of them. This is just kind of shows you that it's varied and it can cause a lot of problems. We're now going to switch to viruses. And viruses, again, different shapes, different sizes, different genetic materials that they're made of. They're very diverse also. It's one of the simplest germs, and there is kind of a debate of whether it's alive or not. But I found this one reference that caused it, that said that viruses are part of the continuum between chemistry and biology. And I really like that because it kind of tells you where they are in the world. They are not really a cell. They can't live alone. They can't do anything without being inside of a cell. I will tell you that there's tons of them out there. There's about 5,000 species that have been identified, but it's estimated that there may be over 1 million species, even up to 100 million different types of species of viruses. And they do that through DNA typing. So just so you know, this virus, this is an electron micrograph of a varicella zoster virus, and varicella zoster produces either chickenpox or shingles, right? And then over here you see this EM micrograph is actually herpes virus, and it's attaching onto a tissue. This is some sort of human tissue. So this is kind of gives us an idea of what a virus looks like. Again, very simple. So you have the genetic material inside, and then you kind of have this envelope that goes around it that's made up of protein. And then you have these glycoproteins, which are a combination of sugar and protein together. They're kind of sticky. I think of them as Velcro again, and they help the virus to stick to whatever it is it wants to either invade or attach onto and cause problems. The virus has to have a host to replicate. It has to use and I'll use a human cell as an example. It has to use a human cell machinery in order to do what it's supposed to do. And its primary function in life is to reproduce. That's all it does, really, in the simplest form, right? So it needs cells to do that. And it does that one of two ways. It causes damage of one of two ways. It either can destroy the host cell or it can damage it. So this cartoon down here is showing how it destroys the cell. So here's our virus. It lands on the host. It sticks onto it. This virus will actually dump its DNA into the host cell. It ends up in the machinery and produces more virus. So it becomes this little factory of virus-producing a virus-producing cell. Once everything is put together, it breaks open that cell, it lyses it and kills it. Another way, and this is influenza that does it this way, and actually coronavirus is similar to this in some ways. It attaches onto the cell pulls it inside where it kind of comes apart. The nucleic acid, the genetic material, goes into this cell machinery. That doesn't happen with coronavirus, but it does with influenza. And it actually starts to, again, turn the cell into a little factory that produces more virus. And in this case, it does what's called budding. It pulls itself out and it takes along some of the cell with it. and eventually this cell is going to die. So here's a whole bunch of different viral infections that we can get. And again, this just kind of gives you the scope of what it is. Unlike bacteria, which are mostly not harmful, for viruses, there aren't a lot of them that are symbiotic with us or maybe even inert with us. Most of them will cause some problem, and that's because they need to fulfill their function to reproduce. So now I'm going to kind of switch gears a little bit, because once you have one of these germs in you, how do you know? How do you diagnose an infection, and how do you know what it is? So again, I'm going to give you guys a poll question. Which of the following indicates that there's an active infection? So the poll is open. You all can choose which one of these you think is the answer. Testing positive for the pathogen. We hear cases all the time. The appearance of symptoms and signs of the disease. So your body is reacting to the presence of the pathogen. You have germs in your body, or the presence of antibodies to the pathogen, or all the above. So let's see here. How are we doing, guys? Okay. This was okay. So Gail's gonna close it. You can see the results. Most of you chose all the above. Sorry, but the actual answer is the appearance of symptoms and signs of the disease. And I'll explain why all of these don't indicate an active infection. I'm not going to do that now. I'm going to do it as we go through. So what is an infection anyway? Remember that we are in constant contact with pathogens, with microbes, with germs, right? But it's really rare that they cause damage, because the host immune responds and is often very successful at beating this back. This is why you hear, of somebody who is testing positive but may not have all the symptoms, because their immune system is fighting this thing, right? You also have the normal microbiome, the fauna and that live in us that help to prevent colonization. And we'll talk a little bit about immune response in the next few slides. So something can colonize us. It can actually be on us and living and all that kind of stuff, and not actually do any harm at all. In order for there to be a disease, there has to be a number of steps that will eventually cause that microbe to make us ill. So it has to, first of all, reach its target site in the body. It has to get to where it needs to be, right? So it then has to attach to the target site so that it's not dislodged, so that the immune system doesn't destroy it. It has to evade the immune system. And then it has to multiply really quickly. So it has to be very fast. Again, it has to beat out that immune system, because at the same time, the immune system is also developing all of its defenses. And then in order for the disease to actually happen, it has to obtain nutrients in order to continue living, and then continue to avoid the immune system of the body. And then you're going to have some sort of damage. And that's really when you have an active infection. So infection doesn't necessarily mean you have disease. It just means that the pathogen is there. And I'll give you an example. If you have certain bacteria in your urinary tract, but you're not having any symptoms of it, you could say that you have a UTI, but the reality is they're not doing anything. And so you don't need to take an antibiotic. You don't need to destroy them, because in that case, the treatment can be worse than the actual presence of those bacteria. There are some bacteria, as you get older, lactobacillus, who are very commonly found in the bladder of older people. They don't do anything. They don't hurt you. And the pathogen is always under attack by not just the immune system, but also the environment. So how do we diagnose an infection? We look for symptoms. maybe a physical exam. The doctor may look at your risk factors, and he also will rule out other disease processes. You might be having problems, breathing. Is that because of an infection, or is there something else going on in your lungs? Like maybe you have COPD or something like that. So the other thing you do to diagnose is to figure out who it is that's causing the problem, right? So you're going to maybe look under a microscope at what that microbe is. You might do a culture. That's the gold standard, because that way you know what it is and that it's actually alive and growing. You could do an antibody test, an antigen test, test for genetic material, PCR, we'll talk about those, or non-nucleic acid-based identification tests. Those aren't done a lot. They're really looking at unusual things. So this, of course, is the coronavirus. So let's talk a little bit about diagnosing individual types of things. So the yeast and fungus infections, the doctor will do a visual inspection of you. Remember, these are a lot of times surface things. So they'll look at your feet, they'll look at your tongue, they'll look at your skin, whatever it is. They might do a microscopic inspection where they actually scrape stuff off and look at it under a microscope to try and identify what kind of fungus it is. They may do a culture, and so over here we see a picture of cultures, 2 different fungal cultures. The problem with cultures is they take time. Fungal cultures can take up to 24 days before you actually can figure out what the fungus is. And then to treat a fungal infection, you would use an antifungal drug. Now, there are only 3 that are approved in the US, and one of the problems is that they're starting to see resistance of fungus to those 3 antifungal drugs. How do you diagnose a bacterial infection? Well, first you want to know whether it's primary or secondary. Primary means that it's the bacteria that's causing the problem. Secondary means that the bacteria has become opportunistic. So maybe you have a viral infection or a fungal infection and the bacteria says, hey, wow, the door's open, I'm coming in. And so how you can tell by symptoms is that often the illness will last longer than the expected viral time, or your fever may be higher than what you expect from just that viral infection, or rather than getting better you get worse. Or common examples of secondary bacterial infections would be sinusitis or ear infections or pneumonias, very common. Another way to look at bacterial infections is to do a blood test and look at white blood cells. White blood cells are part of the immune system. So if the immune system is activated, the number of white blood cells will go up. Now, it's not, the be all and end all, because that could also indicate that you have inflammation. It could show that you have an immune system disorder, or you may have a bone marrow disease. Those are all could be indicative of a high white count could be indicative of all of those. So, they do more than one test is what I'm trying to say. You also want to do a culture and sensitivity, especially if you're going to get an antibiotic. And that's what this is over here. So basically, what you see is this kind of opaque, it's a lawn of bacteria on an agar plate. And then each of these white dots is a little piece of a type of paper that has been dipped into, impregnated with antibiotics, different antibiotics. And so when you lay it down on that lawn, what you see is that this open space is where the antibiotic has killed that lawn of bacteria. Think about like your dog peeing on the yard, right? It kills that space. So these antibiotics aren't that great. This one's really good. These are really good, right? So the doc would pick one of these, maybe this one, as long as you aren't allergic to it or haven't had a reaction to it. This is why you want to do multiple of them, so you have options, right? So how do you treat a bacterial infection? Well, you could wait and see, because often they'll clear, your immune system will clear it. You want to treat the symptoms, of course, so that you're comfortable and just kind of let it go, providing that it's not overwhelming to you. You can take antibiotics. Of course, when you do that, you want to, be really careful that you're not that you're cognizant of your microbiome, that maybe you're doing some probiotics along with it. There's some research on just probiotics being used to fight bacterial infections, because the probiotics really are good bacteria, good microbes that can outcompete against the pathogen. They also help to stimulate the immune system. They can improve your gut health, and they can reduce the side effects of the antibiotic. So they can generally either help with that, or even maybe take over instead of taking antibiotics. But yeah, anything you do, you want to make sure you talk to your doc first, right? Diagnosing viral infections, again, you're looking at symptoms. You can culture them. That's what you see here. So you grow cells in these dishes, and this time maybe, you know, a human cell culture, and then you add the virus to it. And so here we're diluting it out. So here there's a lot of virus. You see it's killed all the cells. This has been stained. And here there's only a few. It also helps you how many. It gives you a number of viruses that are in you, and it also tells you that the viruses are truly alive. You can also do histology, where you're taking a sample of the tissue that might be infected. That's what you see here in this picture. And you can see the type of virus it is. This is actually a MERS virus. This is Middle Eastern Respiratory Syndrome coronavirus. So it's, remember a few years ago we had MERS come through, and that's what this looks like. You can also do antigen detection or nucleic acid detection. So I'm going to talk about nucleic acid detection. In this case, it's PCR. And I'm going to use RNA as my example because this is, you know, this is in the news. This is coronavirus, right? So you take a little piece of this, the mirror image called a primer of the RNA of the, we'll say coronavirus in this case, and the PCR machine and the PCR system is able to then fill this in and make DNA out of it, which is called copy DNA. So it's a mirror image of this RNA. Now we can they designed primers or little pieces of DNA that are very specific to the pathogen, in this case, the coronavirus. And the PCR machine will fill in here and produce a piece that looks like this. So now you actually have two copies, right? Every time you do a cycle, it's increasing the copies. So you're going to do this over and over again, and before you know it, you have enough that you can see. Sorry, I went too fast, but now you can visualize. PCR is, I'm going to go back up to PCR. PCR is actually looking at genetic material. It is, you need specialized equipment, so that's kind of why sometimes it ends up being delayed. And it's very sensitive. Theoretically, you can detect one piece of nucleic acid, so one piece of RNA, one virus using PCR. There's also antigen testing. This is a dipstick. This is, you know, if you've ever seen the pregnancy tests, this is kind of what this is. So you have an antibody to a specific antigen, and an antigen is just a protein that is found on the germs outside, and so you have an antibody to it that has a color label on it. When you dip this in, this complex of antibody and color moves up the strip. When it hits the antigen to the antigen of the bacteria that's stuck on, or whatever the microbe is that's stuck on there, it'll react and cause a color reaction. So here's your positive. This second one is actually a negative antigen, but what it does is it just reacts with the color. So it gives you an idea that the system is actually working, but it has nothing to do with the microbe at all. So basically this tells you you have a positive result, and this tells you it's a negative result, but the antigen dipstick is actually working. It is very fast. It is portable. You don't need special equipment. Positive results are very accurate, but the problem is that negative results might have to be confirmed. It's not as sensitive as PCR. You need more than just one for this to work. Antibody or serology testing. So this is where those of you who said all the above, serology testing or antibody testing is really looking at past infections because the antibody is already produced. It's really telling you that the antibodies are already present. Antibodies are found in blood, so this is a blood test, and it kind of looks like this. Again, you're looking for a color reaction, so no color would be negative. Darker color, these are just diluted out, so you can see that these are all positive. So I'm going to touch a little bit on immunology. I know we only have a few minutes. So I'm going to skip this. I will tell you that all of these are involved in the immune system. So immunity is really resistance to infectious diseases. And our immune system has two pieces, innate and adaptive. So the innate is something that we have right from birth. It's really, it's very immediate, it's non-specific, and it consists of a whole bunch of different things. So including your skin, the barriers, your mucosa, right? Your genetics are part of your innate system, your microbiome. Also, your tissues produce bactericides, something called complement, that will poison the bacteria that are coming in. Inflammation is part of the innate system, and phagocytosis. And this uses cells that are not, they recognize self versus non-self. If it's non-self, they go in there and they basically destroy. And this is what a phagocyte looks like. They're pretty cool looking. It's a very complex system, the innate system. So now I'm going to spend some time. No, I'm just kidding. I'm not spending time on this. This is just to show you how complex this is. The immune system itself is extremely complex. The other half of our immune system is adaptive or acquired immunity. So when the innate system is not enough, then this secondary system, this adaptive system, kicks in. This takes longer because it's very specific to whatever the microorganism or even parts of the microorganism are. It also has memory. So once it is introduced to a pathogen, it kind of remembers it. And that varies depending on what the pathogen is and all sorts of other stuff. But our immune system has a big diversity of antigens in our memory. So which is why, you know, if you had chicken pox when you were a kid, well, that's probably a bad example because you can still get shingles, but you're not going to get chicken pox again. So it's why we get vaccines, because vaccines are really artificial adaptive immunity. So this kind of gives us an idea. There's different parts of it. This is the antibody producing part. And this is the killer T cells. So these are cells that actually go in and will kill the cells that, say, the virus or whatever pathogen it is that's inside the cell. It recognizes it as self or non-self. And this is that macrophage. You can see them sucking stuff in and basically destroying it. So that's what their function is. Pretty cool stuff. Oh, there's my macrophage, and this is just a cartoon of it. So this is a summary of our defenses, innate and adaptive. And then I just want to take a little bit of time and talk about vaccines. It's really immunity without being sick. And so what ends up happening is something of the pathogen is put in us, either a dead pathogen or a less potent relative or parts of that pathogen. And then our body responds using the adaptive immune response. And so next time we see that pathogen, our immune system's ready to go. And it's really impacts individual and herd immunity. So herd immunity really is how vaccines work. So vaccines help us as an individual, but they also help those around us. So if you look at this, you see that some people maybe cannot take the vaccine. So this guy here can't take a vaccine for whatever reason. If everybody around him takes the vaccine, they can't get the disease, so they can't spread it to him, right? So that's the basis of herd immunity. So how do we improve our immune system? Well, we want to take care of our best supporting immune organ, which is the microbiome. And I told you how we have the GI, the gastrointestinal microbiome, And we also have our skin microbiome. It's really the genetic material of all the microbes in us and on us. And they help with a whole bunch of stuff, but they really help to regulate that immune system and help to prime it. It's really unique to individuals. So this kind of gives you an idea of the kinds of things the microbiome does. What ends up happening is there's kind of this symbiosis between the bacteria within our GI system, within our gut, and it helps with checks and balances for the immune system. And if that gets upset, like antibiotics, right, which mess up everything, not that I'm saying you shouldn't take antibiotics, please don't get me wrong, just don't overtake them, but it increases risks for certain diseases. Including autoimmune diseases. And the other thing that's happening is, I don't know about you, but when I was a kid, we went outside, we played in the mud, we probably ate some of the mud, and we really were exposed to a lot of the environment and a lot of different microorganisms. Remember, there's over 1 billion of them in a teaspoon of dirt. Kids now aren't going out as much. They're not as interactive with the environment. And because of that, they have more autoimmune diseases, they have more allergies. So how can you help with that? Get your kids out, get a dog. It can help prevent asthma in kids. Can you improve your immune system? Well, sort of. I mean, really, it's a balance. So living a healthy lifestyle, really important. All those things that we tell you to do all the time, you know, eat your veggies, exercise, adequate sleep, minimize stress, wash your hands, all that stuff is going to help with your immune system. Do supplements work? There's not a lot of hard evidence that supplements work. But if you are going to try a supplement, then make sure you talk to your doctor first, because they can interact with medications that you're taking. As you get older, your immune system just really naturally starts to decline. There's a lot of reasons for that. What ends up happening, the result is you have more infections, your infections are more severe, you have more cancer. Vaccines are less efficient, but they'll attenuate the disease. So maybe they won't stop it completely, but it makes it less worse, less deadly. And there's actually a link to micronutrient deficiency and age and healthy immune. So things like zinc, selenium, iron, copper, folic acid, vitamins A, B6, C, and E might be deficient in older people. So those are some things you may want to look into. So summary, you know, there's a lot of germs out there. Some of them are beneficial or inert, or actually most of them are. There are some that can cause infectious diseases in us. You know, don't be afraid of vaccines. They're very safe. They're highly tested. You know, they can prevent infectious diseases. They help us individually. They help others. And keeping a healthy immune system can help to defeat those infectious diseases. And really, it's all about healthy lifestyle. I apologize for going over. Oops, sorry, I went a little too far. If anybody has any questions, I am happy to stay on and answer those questions.
Well, Linda, we just have a few questions. Okay. Thank you. That was very, very informative. And luckily, everyone's going to get a recording so they can go back over that. There was just so much information, but everybody a little bit later. If you have to leave now because you only budgeted an hour for this, we understand. And when you leave, you'll receive a 5-question survey. We asked you to look at that. And also you'll get information about our upcoming financial symposium, which is going to be January 26th and 27th. But otherwise, if you can stay on for just a few more minutes, we just have a couple questions that are very interesting. Okay, so the first one is, how do viruses mutate and why?
So now you're making me go back into my virology classes, which were, I won't tell you how long ago, but pretty long ago. You know, let me, so viruses, why do they mutate? Well, remember their main function is to survive and replicate. And so really the way they do that is to be able to change how they are and what they do. And so, you know, this is kind of why they mutate. So do they mutate readily? More than bacteria? Probably not, actually, because bacteria can really exchange a lot of genetic material among themselves and even into the human genome. It's kind of interesting because viruses also, through our evolution, have integrated into our genetic makeup, which is kind of interesting. So even though we say that most viruses are harmful, we have a lot of viral material inside of us. So that's kind of its separate thing. Honestly, I'd have to look up the specifics of that. So I'm going to kind of table that. But what I'll do is I'll find out that answer, and I will make sure it gets back to everybody. I'll get Gail to email it out to everybody.
Thank you, Linda. Next question is, this person's understanding is that the 2 vaccines against the coronavirus do not actually prevent the infection, but prevent the symptoms. Is this correct? And if so, how would herd immunity occur?
So I'm not sure that's correct or not. I'm actually going to move on because I actually had some, I want to show you, because these vaccines are mRNA vaccines, which are a little different than what we would consider like normal vaccines. Like the flu vaccine is different. These are the flu vaccine. They use a attenuated or a dead virus, and they inject it into you, and it causes an immune response. This, because these guys are RNA, and I'll skip this, but basically what ends up happening is that mRNA, it gets into the human cell and it produces the protein, the virus protein that's on the outside of that virus, of the coronavirus. And then the cell kind of shifts that outside, and it produces an immune response to that antigen, which is the protein. So it is producing an immune response. It's producing an antibody to the virus protein. So I'm not sure, why it wouldn't then cause you to not be able to get that virus. I know there's a lot of misinformation out there. This information I got right off of the CDC. So I'm not sure I answered that or not.
Well, it's very confusing, and I think they're still trying to figure out like how this works.
The mRNA vaccines are brand new. They've not been used before. They've been working on them for a long time, but they have not been used before. And so there is a lot of confusion as to what's going on with it. I do know that, it doesn't come into I think maybe the confusion is that the RNA normally in the virus goes into the cell and is used by the genetic machinery of the cell. But the vaccine, it never goes into the genetic material of the cell. It stays out in where the rest of the factory is that's producing proteins. So that might be some of the confusion.
Okay, and do you know if those, do they know yet if those who've recovered from COVID still need to get a vaccination?
I really try to avoid any coronavirus stuff at all because there's so much information out there. So I'll tell you, I've read that they're saying, yes, you should get the vaccine. I will tell you my opinion. To me, it's not logical because if you already have the antibodies to it, why would you need to get a vaccine? It kind of doesn't, I don't know. I don't know the answer to that question. I'm not sure anybody knows, because like I said, there's a lot of information out there. Unfortunately, not all of it is accurate, and we may not know for a few years as to what's going on. There's a lot of information on whether your immune system, whether you're immune to a coronavirus after you've had it, for how long you're immune. I mean, there's just a lot of stuff out there. Can you get it again? Probably. I mean, most viral infections you can get again. I had chickenpox twice. you know, so it's not like it's unheard of. Is it normal? I mean, is it usual? It's rare. It's rare. So, but you know, I'm not a coronavirus expert, so.
Right. Thank you. Okay, so here's a question. Someone said they were told in the past that they could not donate blood for a specific need for a coworker's son because they were carrying the cytomegalovirus. I hope I'm pronouncing that right. And can you explain why?
Everybody's asking me virus questions. No, I don't know. I honestly, my past, I'll tell you, I did a little, I mean, this nanometer amounts of virology. It's mostly bacteriology that I worked on. So does anybody have a bacteria question? No, just kidding. I don't know. I mean, cytomegalovirus, from what I remember, will cause disease. And so it's probably one of those things that is, once you have it, it's in your system and, you know, they don't want it to pass on to anybody else. That would be my guess, you know, and it probably, I'm guessing again, that it's associated with the bloodstream because otherwise, like you think about VZV, which causes chickenpox and shingles, it actually hangs out in the nerves. And so, you know, you could give blood, you're not going to get it from somebody who has that already in them.
All right, so next question is, do you know why some antibodies are permanent in the body while others diminish?
Antibodies. So the antibodies themselves, the actual antibodies, just to clear this up, don't actually stay forever. So there are actually cells called memory cells that basically have, I'll say, the blueprint of those antibodies so that when that pathogen comes in, a whole new batch of antibodies are produced. So usually what ends up happening is it's not like, so it's not that necessarily that the antibody, the memory cell goes away. It's that the pathogen changes. So for example, influenza. So why do we have to get influenza vaccines every year? It's because the influenza virus changes every year. It mutates. And part of that is to be able to evade the immune system. You know, it's not like it's thinking, but it just, you know, the ones that are able to do that survive. And so it's survival of the fittest in this case. And so now you have a different kind. It's slightly different. You know, when we make antibodies, you think of them as a Y, and that piece in between is very specific. You know, that Y, that juncture is very specific to a particular antigen. Think of it as a lock and a key. Not every key will fit every lock. So if your influenza is a certain key and you have the right lock for it, when that influenza comes in, your body will be able to unlock the immune response. If all of a sudden, you know, you have this lock, but the influenza has changed the key structure, it's no longer going to work. And so it's no longer going to produce antibodies to that specific. And so the body, you know, is not going to see it and you're going to end up with influenza. That's why we have different vaccines every year. I hope that answered that.
Yeah, I think that's a great answer. Thank you. Thank you very much. And we just have one question left.
Okay.
Can you tell us about like the capsules with the healthy fecal matter that are being used to help people with their gut health?
Yeah, so when we talk about the microbiome and in the gut, it's kind of interesting because I mentioned that there are these pathogens that will kind of take over. Say, for example, if you take antibiotics, you might have gotten, or maybe somebody you know has gotten C. difficile, which is a bacteria. And what ends up happening is C. difficile is normally in your gut. But what happens when you take antibiotics is the antibiotics destroy everybody else, and the C. difficile says, "Whoa, party in the gut now," and they grow like crazy, and they cause this, you know, diarrhea and upset stomach and all that kind of stuff, right? So it can be very devastating, especially for older people. So what ends up happening is if the doctors can't get rid of that bacteria, one way to get rid of it is to restore the normal bacteria, different kinds of bacteria that live in the gut. And so what they have done is they've actually been able to isolate the bacteria out of feces. I know this sounds really gross, but they're isolated. Everything's cleaned up. Everything's, pure. There's no fecal matter in there. It's just the bacteria, and they're able to capsule them and make them into a kind of probiotic that when you take that, it restores the normal microbiome, the normal flora and fauna that live in your gut system, and it gets rid of the C. diff. It's amazing, and it doesn't come back. I mean, it's really a great cure. There's been pushback on it because of it coming from fecal matter. You can kind of understand that, but it's really great, and it's very clean. It's not like you're, eating poop. It's really a good system.
That's a very creative solution, isn't it?
It is.
And thank you so much for you for all the work that you put into this. I really appreciate it. That was a very thorough presentation, and I think we all have a better understanding of all the different microorganisms now. And thank you to everyone for listening today. We appreciate it. We hope that everyone has a wonderful holiday season. So thank you very much to everyone. Have a great day. Take care.
Thank you. Bye-bye.
Bye-bye.